The next pathway, if you just look at this, this should look very simple to you. This is the classical pathway, okay? And this pathway, again, as I told you that there are common things in the lectin pathway and the classical pathway. This pathway, is activated by some immunoglobulins like the IgM or IgG immunoglobulins, particularly the subclass 4, IgG4 immunoglobulins can activate this particular pathway. And this pathway can also be activated by the C-reactive protein. Okay, these are acute phase proteins which are synthesized in the liver when there is some kind of infection. So these proteins can also activate this particular classical pathway. This is another way of complement activation. Okay, and how does it happens here? If you see here, this is a C-reactive protein, okay? And where is the C-reactive protein, bonded to? The C-reactive protein is bonded to the pathogen because the pathogen has infected a person, and as a result of that infection, C-reactive proteins have been synthesized, and they target the pathogens directly. So they have bound now to the surface of the pathogen. Now here are IgM or IgG antibodies, as you see here. They are also bound to the surface of the pathogen, as you see here. Okay, so this is an antigen, and these are immunoglobulins. Where do immunoglobulins come from? How the immunoglobulins are synthesized in your body? Do you know that? You know about adaptive immune responses. When there is infection, of course the infection ultimately is going to lead to antibodies production, specific antibodies production against pathogens. Okay, so immunoglobulins production is going to take place inside your bodies as well. Immunoglobulins are, very specific immunoglobulins in the form of antibodies produced by B cells. These are antibodies which are part of the membrane of the B cells, and you have studied all those antibodies. And there are other types of immunoglobulins. There are 5 different types of immunoglobulins synthesized in our body. The liver also synthesizes different immunoglobulins. And these immunoglobulins, when they are attached to their partner pathogens, which these immunoglobulins recognize, then the later events are recognition by the complement system. Now the complement factors, some of the complement factors like C1 would recognize the C-reactive protein bound to a pathogen, the surface of a pathogen. Or the C1 complement can also recognize an immunoglobulin, whether this is IgM or IgG. IgM antibodies are secreted during the initial phase of infections. Both these assemblies are perfect invitation for the complement factor 1, which is already present there inside the body. Okay, so the complement factor 1, when it finds such kind of combinations, it is activated. And how? C1 again is a combination of different small peptides, which include C1R, C1S, and C1Q. Okay, and these peptides would join together to make a single complement, which is known as C1, complement 1. So complement 1 is a combination of these smaller peptides or smaller portion. C1Q, this part binds to the FC region. FC region, where is the FC region? In immunoglobulins. Okay, so FC region and activates C1R and C1S. Once this assembly of the C1Q with these particular factors, IgM or IgG or C-reactive protein on the surface of pathogen would take place. So after that, what will happen? The C1Q, this part would start activation of the C1R and C1S, okay? And C1S is the first to be activated as a result of binding of C1Q either with C-reactive protein or with IgM or IgG antibodies, and this activation of the C1S would lead to activation of the C1R would be activated first, which would lead to activation of C1S, and the C1S, when activated in this assembly, would go for attracting factors like C4. Just like in the previous slide you saw in lectin pathway that C4 was deceived by the assembly of lectin in mannose, which and the MASP or serine proteases, that was the assembly there, which attracted this particular factor C4. But here in this assembly, as you see, there is a single complement with three different parts, one part activating the other part in a sequence as a result of recognizing this assembly. So C1Q first binds with this assembly, activating C1R, and C1R then activates C1S, and the C1S then cleaves the complement number 4 into 4A and 4B. And similarly, as it happened in the previous lectin pathway, C4B is the heavier part. It is heavy, so it falls down on the ground. Okay, what is the ground? This is the pathogen surface, and it just fixes to the pathogen surface, making a covalent linkage just like the C3b molecules would do. Okay, now this happens the same way in case of the C-reactive protein and the IgM or IgG antibodies, both the molecules that can activate this particular classical complement pathway. The rest of the events are the same in lectin pathway and in the classical pathway. Okay, as you would see in the next slide, these things happens in both of these pathways. Okay, so here they are activated MASP2 cleaves C4 into two parts, as you see here, the 4A and 4B. 4B just binds to the surface, then MASP2 cleaves another complement which is C2, and C2 is cleaved. The bigger part just binds to C4B, and this C4B and C2A, this is the convertase enzymes formed in both these cases. And what is it going to do? It's going to cleave the complement number 4 and number 3 into 3A and 3B. Okay, and finally the 3B fixation takes place on the surface of the pathogen, and I told you about the importance of this fixation. Okay, C3b can mark the pathogens for phagocytosis, and C3b can also lead to the formation of membrane attack complex, as you saw in the alternate pathway. So these are the events actually that are taking place in the classical or lectin pathway. Okay, and now we just had to go through some lessons that we have learned during discussions on the complement system. Okay, so this is some poetry that you can enjoy after you go through this lesson. Okay, this is now everybody is activated. When they saw the poetry, it's okay. The boring discussion will come to an end in a while. Okay, what are the lessons learned? Well, after we discussed these different complements in this chapter, the lesson learned, the major lessons or major terminologies that you should keep in mind are these. Okay, also for the paper, the upcoming midterm, which is scheduled for 16. Okay, the complement system is a set of serum proteins that coat the surface of the pathogens, making them more readily phagocytosed, are inducing lysis via the membrane attack complex. These are the two modes of complements, that how the complements are going to destroy the pathogens. Okay, either they mark them or they make a membrane attack complex and just lyse the cell. There are three complement pathways that act at different times and are initiated by distinct mechanisms. And you saw that the alternate pathway, the lectin and the classical pathway, are initiated in different ways by different mechanisms. Okay, so we discussed in detail that alternate pathway is the principal pathway, but there are some pathogens that can be recognized by other ways as well. And the classical and lectin pathway are doing the same, that particular job. The cleavage of C3, while C3 convertase into C3A and C3B is critical step in the complement fixation. Okay, C3 is the most important complement. And I told you about the abundance of C3 in body fluids. 1.2 milligram per ml of your body fluids is C3. So you can just imagine about the concentration of this complement. C3b codes pathogens for destruction, okay, and complement receptor number 1 on macrophages and other phagocytic cells recognizes C3b coded pathogens and promotes phagocytosis of the pathogens. We also have discussed this particular part. But CR1 is not the only receptors on macrophages for identification of pathogens. We also have discussed these factors. C3A and C5A can recruit immune cells to the site of infection. Okay, and somebody who is doing recruitment is going to attract these cells somehow by some mechanism. And we also have discussed these mechanisms during our discussions on these chapters. They are also anaphylactoxins because they can induce anaphylactic shock when an acute inflammatory response occurs throughout the body. Okay, anaphylaxis are hypersensitivity reactions that these complements can cause sometimes inside the bodies. So complement is a set of plasma proteins that act on a cascade of in a cascade of reactions to attack extracellular forms of pathogens. Complement fixation, the attachment of C3b to the pathogen surface, which labels the pathogen for phagocytosis. Classical complement pathway, the complement pathway initiated by the antibody or C-reactive protein bound to the pathogen, inducing C1, C4, and C2 to generate C3 and C5 convertases. The formation of C5 convertase would be the same way as it happened in alternative pathway. Okay? The only differences among the pathways were those that we discussed today. Alternative complement pathway, the complement pathway initiated by the presence of a pathogen that induces the cleavage of C3. So it's directly going to cleave C3 into C3A and B. So this is the easiest way for the body to adapt to and convert C3, which is the most abundant complement, to fix it on the surface of pathogens. Lectin complement pathway, we just discussed this lectin complement pathway, right? Then C3 convertase, you also know about the composition of C3 convertase in alternate pathway and in the classical and lectin pathways, that there are differences, okay? But all C3 convertases would convert C3 into a bigger and smaller part. They would just cleave C3. Whether the composition is C4B, 2A, or whether the composition is some other composition. C5 convertase, complexes that cleave complement component 5, it is a convertase that cleaves C5 into a smaller and bigger fragment, inactivated. Complement receptor, you know about it. Opsonization is the coding of the pathogen, you know about it. Membrane attack complex is the complex made as a result of complement activation, which can lead to opsonization or formation of the membrane attack complex, which would include complements from C5 to C9. Okay, and I also have told you about the regulation of these, complement, the membrane attack complex, for instance, or the regulation of the complements. So, without an opsonant such as an antibody, the negatively charged cell walls of the pathogen and phagocytes repel each other. Okay? Why opsonization increases phagocytosis, if somebody asks you? So this is the answer to that. Okay? The pathogen can then avoid destruction and continue to replicate inside the human body. Okay? So without an opsonant, if there is no opsonant, such as an antibody marking, antibodies also used to mark cells. Complements are also used to mark cells. Okay? In that case, if there are no opsonants, then the negatively charged cell walls of the pathogen and phagocyte repel each other. Okay? And in that case, the pathogen can then avoid destruction and it would continue replication inside the cell and thus doing damage to the cell. So that's why it is important. You know about antibody. These are protein secreted from plasma cells like antibody producing B cells that binds antigens, proteins or peptides recognized by the adaptive immune cells. C-reactive proteins, soluble acute phase proteins that binds phosphocholine on bacteria, making them more susceptible to phagocytosis, either directly or through the classical complement pathway. Okay, so C-reactive protein is also used for labeling the pathogens inside the body. You should also, you should remember it, just like antibodies, or the complement factors, C-reactive protein can also do the same thing. Then anaphylatoxins, you know about C3A and C5A, that they can act as anaphylatoxins and chemoattractants. Okay, and they enhance this process of inflammation. They recruit more immune cells to the site of infection, and they can also cause hypersensitivity reactions. They are involved in that process as well, because there are receptors on different immune cells for C5A and C3A. For instance, the mast cells have got receptors, for C3A and C5A. Receptor means that the mast cells can be activated by C3A and C5A. Okay? If there is this smaller complement of the complement number 3 or the complement number 5 in the surrounding of the mast cells are abundant under your skin. Okay? So these complements can activate the mast cells, and the result of activation of the mast cell is very, dangerous. In hypersensitivity reactions, for instance, they can just, release very toxic granules in the surrounding tissues, and these granules contain very toxic or hydrolytic enzymes. And when the tissues or cells are exposed to these hydrolytic enzymes, they create this very dangerous or very, bad inflammatory situation in the surroundings. Okay, so these factors, they are involved in doing different jobs, including these anaphylaxis or hypersensitivity reactions by influencing or activating some of the immune cells like the mast cells. Mannose binding lectin, this is also a soluble acute phase protein in the blood. It binds to mannose residues on the pathogen, as I told you, and activate the complement pathway, as you saw in the lectin pathway. Okay, so now this part is finished, and your midterm exam would include the course that we have studied till now. Okay. Yes.